Good afternoon, this is Group 1, looking at Arabidopsis Thaliana Chromosome 1, region 500 kilobases to 700 kilobases, searching for potential resistance genes.
Overall, in our 200 kb range, we found a total of 65 genes with about 1 every 3 kb, which is a little denser than the general rule of 1 gene per 5 kb. Overall, our segment was packed with genes, SNPs, and transposons, along with many ESTs to support the gene models predicted. 3/4 of these genes were known, and a quarter with only EST data but no function, and one gene which was hypothetical, which means there was no EST to support it and was only predicted via computer. Gene function fell into a large amount of categories, even when broadly defined, with 17% and 15% falling under other and unknown respectively. Other functions in decreasing order were transport, modification, metabolic, anabolic, expression, catabolic, and resistance. Resistance, even though had the lowest, was still 7% or 4 genes, which is pretty high considering that we're looking at a region that hopefully had resistance genes, but we didn't know. Box and whisker plots were shown for introns, exons, transposable elements, gene length, and gene models, and how they were distributed amongst the genes. The graphs speak for themselves, with a couple of highlighted notes that our median gene length was about 1,800 base pairs, but did range from as small as 71 base pairs to as large as 7,000 base pairs. There were usually between 3 to 9 tDNA inserts found, along with 2 to 9 introns for each gene. Pretty much only one gene model was seen for each gene, though one gene had as many as 5. A phylogenetic tree was built using Cluster Omega, the new version of Cluster W, using the multiple sequence alignment for our cluster seen here. We see that 2940 and 2950 have the same common ancestor and therefore cannot be concluded which one came first. 2920 would have come next based on this tree. We can see that the gene from our cluster 2930 and 2750, our picked resistance gene, were equally related to the rest of the genes, and based on this tree would not have been able to tell which one diverged first. Since 2920 and 2930 are resistance genes, it makes sense that they are more closely related to the resistance gene 2750 than 2940 and 2950, which do not confer resistance.
Now, to investigate these genes individually, we will look at the chromosome one gene at a time.
It is important to note that in our Prezi, we refer to the number of gene models as alternative splicing before we figured out how the correct nomenclature was gene models. So when it says alt splice, it means gene models.
Our first cluster is 2 genes. They're reciprocal best blast hits to one another. They are polyketide cyclase, dehydrase, and lipid transport genes. They have respectively 3 and 7 tDNA inserts, and their lengths are 1,859 base pairs and 1,561 base pairs. Their biological function is unknown, although they are involved in the creation of some type of polyketide and opening the guard cells. They're expressed in the chloroplast of the plant during the C globular stage, petal differentiation, and expansion stage, and they are expressed in the tissues of the carpel, leaf structure, flower, guard cell, pedicel, petal, plant embryo, pollen, sepal, stamen, and stem. They also have poor blast hits to a phosphatase gene involved in cell signaling, a kinesin gene involved in movement, and a guard cell ubiquitin ligase. Zooming into this cluster, we can see that 2470 has 3 tDNA inserts, one of which is the SALK 123221. You can see its flanking sequence here. which we could use if we want to investigate this gene's function further.
This gene codes for a tRNA, which is a transfer RNA, not a transposon. It is the RNA sequence that has the antisense region for the amino acid which is used to build the polypeptide. tRNAs are typically 70 base pairs long, so they are small molecules with an important function, with multiple copies of each tRNA in the genome depending on the species. In our segment, we have tRNAs for histidine, lysine, phenolphthalein, and possibly another based on a potential tDNA antisense region.
Gene 2510 controls the K+ channel, which involves transport and opening and closing of the stomata. There are 4 hydrolase genes, which are in different supergroup families due to slightly different function, but can be considered a cluster because they are all hydrolases. These genes are AT1G02640, 2660, 2670, and 2800. So they're not located exactly right next to each other, but are really close to one another within the genome. All but the third gene are biologically involved in some sort of metabolic process, with the first one and the last one being involved in carbohydrate metabolic process, and the second one in the lipid metabolic process. The third gene, however, is involved in DNA repair. There are a total of 39 transposons between all 4 genes, which is about 10 transposons per gene. And there's 1 transcript per gene, and about 34 exons and 30 total introns throughout the entire cluster, and an average gene length of 3,454. And I'm looking specifically at the promoter sequences. 69 of them are found in all 4 genes, while the rest are found in at least 3 of the genes, while doing a motif analysis of the promoter.
The resistance gene that we picked was AT1G02750, with a length of about 1,800 base pairs, 3 introns, 4 exons, 2 gene models, as well as 2 tDNA inserts. Its biological process is response to drought and salinity, and is located both intracellular and in the nucleus. Its expression is pretty much in every tissue in every stage of development, so it's a pretty ubiquitous gene. There were 61 known polymorphisms within this gene. We picked 2750 as a possible resistance gene, in part for its decently large amount of information, also for the sudden stop of interest in 2008 as an R gene. And yes, salinity and drought are diseases. They are abiotic diseases, which are just as important as biotic diseases in the amount of damage done and being able to confer resistance against. It is referenced in multiple papers as ATDI19, which is Arabidopsis Thaliana drought-induced gene 19. This ABA insensitive gene family is induced when salt is present.
Looking at the methylation patterns for this region, we see that this gene is mostly unmethylated, demonstrating that it is under high expression. Gene 2800 is the last of the hydrolase cluster discussed previously.
So this is the first of a cluster of 2 ubiquitin ligases. The first gene has 2 alternative transcripts. There's 11 total transposons between the 2 genes. There are approximately 10 exons and 9 introns per gene, and both have a molecular process of ubiquitin protein ligase binding, with the first gene being involved in many biological processes, a few of them being cellular response to phosphate starvation, defense response to bacterium, regulation of metabolic processes, and other responses, while the second gene is only biologically known to be involved in the ubiquitin-dependent protein catabolic process. Remember that ubiquitin marks proteins for degradation and programmed cell death. The largest gene cluster in our region that does not have any other genes interrupting it occurs near the end of our genome region, beginning at 658 kilobase pairs and ending at 666 kilobase pairs. It consists of 4 genes, as they're pictured here, which are located in the glutathione S-transferase family. In total, there are 16 transposons within the cluster. On average, there's only 1 gene product for each gene, while the second has 2 and the fourth has 4 alternative transcripts. The average gene length over the 4 genes is 1185.5, and there's approximately 3.5 exons and 2.5 introns between the 4 genes. In doing a motif analysis of the promoter, all of the gene sequences were found in at least 4 genes, and there were 76 sequences that were found in all 4. And the molecular function of all 4 of these genes is glutathione transferase activity. However, in looking more in depth at the biological process, this differs between the 4 genes. All 4 genes are biologically involved in the toxin catabolic process. However, the first 2 are biologically responsible for defense response to bacterium, response to the cadmium ion, and response to salt stress, while the first gene is also responsible for the defense response to fungus, and the last 2 are only known to be involved in the toxin catabolic process. There is a predicted gene cluster between 2960 and 2965, which are best protein matches of each other, but the biological and molecular function of these genes is unknown. The only thing that we know is that there are blast hits to 99 proteins in the first gene and 8 blast hits to 8 proteins in the second gene.
Gene 2970 is a Wee1 kinase homolog. It's involved in cell cycle progression, serving as a DNA damage checkpoint, preventing mutations that are unfavorable or damaged chromosomes from being inherited in the next generation.
This is the second of the previously discussed cluster of 2 ubiquitin ligases.
This graphic shows the areas of synteny in our region of the Arabidopsis Thaliana Chromosome 1. You can see that the genes in Arabidopsis Thaliana are almost perfectly syntenic with Arabidopsis lyrata, although the genome for lyrata seems to be a little bit more compact, but the gene order is conserved. There are more glaring differences when compared to poplar, with the syntenic blocks being more spaced out, and there's a large region around 600,000 kb in Arabidopsis thaliana that does not show synteny with poplar. Now let's focus on the region containing our resistance cluster. This cluster falls within a syntenic block between Arabidopsis thaliana and lyrata. The genes as well as the intergenic regions are found nearly unchanged between these 2 species. Although there is a synteny block between Arabidopsis thaliana and Populus trichocarpa in this region as well, the region is not nearly as close to an exact copy. It appears that genes 2920 and 2930 in Arabidopsis thaliana are represented by the same gene 0250 in Populus. However, 2940 in Arabidopsis falls in an intergenic region in Populus, which may signify that the gene has lost function. The high volume of inversions in this area between the 2 species support the hypothesis that gene function has been lost in Populus, but more in-depth analysis is required. The Arabidopsis thaliana gene 2950 is almost directly syntenic with the Populus 257 gene, except that the 5' UTR region is separated by a possible 1.5 kilobase insertion in the Populus genome.
In closing, the resistance gene we found for region 500 kb to 700 kb in Arabidopsis Thaliana Chromosome 1 is AT1G02750, offering drought resistance to the plant. As you can see, there are a few tDNA inserts in this region that we may exploit for further analysis of the effects of this gene on the plant.